For a few more people to straggle in, I thought I would take the opportunity to introduce you to the seminar series, not just here, but at UNBC in general. I see mostly familiar faces here, but for those of you who don't know me, my name is Phil Burton. I'm the regional chair for UNBC in the Northwest here and at the Terrace campus. And you have before you on your desks and up on the screen just a listing of the various speakers we have lined up for the rest of the semester here. It's really been my objective to invite researchers who are active in the area and often hiding in the woods, but also professionals that can give our students and community members an appreciation of the many complicated and worthwhile professions that people are practicing in the region. We're obviously kicking off with Dr. Krisinger here today. 2 weeks from now, we have Ann Travers from Simon Fraser University, who will be talking about transgendered children and the families that deal with them and that adjustment in society. Colleen Austin from the local Kitsim-Kalem School will talk about Indigenous language and culture revitalization. Mikkel D'Angeli, who's a sessional instructor currently with UNBC and teaches at that school, will be talking about her challenges as technically an American citizen trying to work on traditional Tsimshian territory in Canada. And in fact, how that's covered not only by treaties, but all sorts of bureaucracies. Then Letitia Kistimus will be talking about the Plastic-Free Terrace Initiative. Richard Schuster is a postdoc at Carleton University who's actually living and working in Terrace, but working on large-scale conservation biology issues. Cedar Welsh is an alumna of UNBC and working on her PhD at UVic. In fact, she used to be a lab instructor, a faculty member here, and she'll be telling us about her research in the Northwest, the history of glacial changes and their impacts to watersheds. Then at the next day, outside of our normal noon hour presentations, the students of Environmental Studies 414 will be engaged in an Oxford-style debate on a topic yet to be determined regarding professional or environmental ethics. And the format for an Oxford-style debate, in fact, is one to invite audience participation, so that should be quite stimulating as well. And then we'll end the season, going well into the final exam schedule actually, with Maureen Atkinson and Ed Harrison, our own UNBC faculty members, talking about their historical research into the community of Georgetown Mills. Now we also have another series of events that we'll be hosting, which is the BC Agriculture and Climate Change Education Series. So this is a series of webinars hosted out of UBC Vancouver, in which we have nodes around the province. So we'll be here on a month of Thursdays starting February 28th, and I'll be in the room as well to facilitate discussion and feedback to the group. So I know many of you are interested in food production, food security issues, and the topic here is how those are going to respond to climate change. For this one, registration is desired, and we still have room for more participants. If you're interested, please email this address or feel free to contact me. And just a bit of context here, the UNBC main campus also has seminar series that are available by live stream. So the Global Fridays happen every Fridays at noon from 12 until 1:30. All sorts of interesting topics that you can listen in on. At the end of the day, at 3:30, the Natural Resources and Environmental Studies Institute likewise has a series of seminars called NRESI Colloquium. They're also available online. In fact, that'll usually direct you to, again, just unbc.ca/livestream. So these are all ways in which UNBC is in the North for the North. Often we are asked, why don't we have engineering programs available here, or why can't you get a business degree in Terrace? And if you have those interests or know people who do have those requests, please have them come and see me. But in the meanwhile, this outreach, this connecting you to the scholarly world of research and investigation, I think is part of UNBC's mandate. So, Al.
I wonder if it might not be too much trouble for one of your employees to send all this by email to the list.
We can have Velma send this to the same list that notified people here today. Sure. Very good. So with that, I'm going to turn you over to John Krisinger, who most of you know as a former West Community College, now Coast Mountain College. John is recently retired, so spending more time traveling and skiing and maintaining the most complete wood pile I've ever seen in the Northwest. He really has that wood dried and ready for at least two winters of severe cold. And John has a background in biochemistry, maybe some cell molecular biology, and has probably been responsible for educating all of our nursing students over the last decade or so, and probably many of them that you bump into at the hospital. So John, I'll let you take it from here. Here is the magic wand.
All right, thank you, Phil. Well, thank you for inviting me. Phil has actually asked me to do something, and is my next slide my first slide up? Yeah. Oh, there it is. So I have been teaching cell biology and molecular biology, genetics over my career, and anyway, I'll show you a little slide here about a textbook I worked through on human evolutionary genetics, and it prompted me to put this lecture together some years ago. And an interesting quote to start with. So this guy, W.S. Gilbert, he said, "Darwinian man, though well behaved, is nothing but a monkey shaved." So obviously you can see what he had in mind. So here's a textbook that I worked through some years ago. It's from 2014, but I added more up-to-date information on this, and the whole book is entitled Human Evolutionary Genetics. And amongst many things it pursues is what makes us human? So what is uniquely separating us from other related species, especially the other primates? So, and as you might be aware of, in 2003, the Human Genome Project was finished. So that means we didn't map the genome, we sequenced it. So we know the entire letters of the DNA alphabet. And so by now, sequencing has become so cheap and so fast. Thousands of organisms, mainly microbes, have been sequenced, but now also a lot of primates, mammals, etc. And also thousands of people's genomes have been sequenced. So what did we learn from this? Now, in the limited time I have, I want to just highlight a few anatomical, physiological differences between us and any other animal, and then the unique mental features that allow us to happen because of our brain differences. Now, our species itself goes back about 6 to 7 million years ago. We had this common ancestor. I hope you all know we didn't descend from monkeys. We have a common ancestor, of course. So, and like in evolution, the case, there's a lot of lineages that go extinct. So when it comes to present extant species, of course, we have a number of primates that are still around. And then we have, of course, only us, and then very recently extinct human hominid species like the Neanderthals. So as we know now by DNA analysis as well, we have sequenced actually 2 fossil creatures, well, not 2 members, multiple, but the Neanderthals. And then recently in 2010, they discovered another lineage of more modern hominids called the Denisovans that were found in a Siberian cave. And we have interbred with them. At times we shared this planet. And in fact, I have myself sequenced by me and 23. I'm highly related to the Neanderthals. And no wonder, I used to live there. The Neanderthal is actually a small river near Düsseldorf and Cologne. And I was born and raised 60 kilometers from there. But I mean, that's just their name. They were all over the place, as you can tell by the blue shaded area. So then our species, somebody eventually decided we call ourselves Homo Sapiens. Biologically, that's not really a clear line. They decided when people develop culture, like when they bury their dead, when they have arts, that sort of indications we call that now is the period of modern Homo Sapiens. And that happened somewhere around 200,000 years ago. And we know all modern humans originally come from an African population. The red arrows show how that species that is still extant, us, migrated and populated Eurasia. And interestingly, they very quickly already found a way to Australia. One advantage was in ice ages, the ocean level was so much lower. There was a lot of islands that they could use to hop with boats and reach such places. Obviously, fairly late, they populated the Americas, about 15,000 years. Those numbers often change somewhat, but roughly that's probably the truth. And then there's some interesting spectacular differences. For example, islands sometimes are very lately discovered by peoples. So Madagascar, and they don't say anything about Hawaii. Hawaii was probably populated, some people say 2,000 years ago, some 1,000, so fairly recently when you consider the whole time frame of evolution. Now, I know many of you are not scientists firm on genetic terms, and we need to deal with this, so I want to quickly give you some definitions. We all know the word gene. What the heck is a gene? So a gene is a structural part of a chromosome, so a small segment, and it has the instructions how to make molecules that then operate in life. It used to be that we thought every gene encodes what eventually becomes a protein in the biochemical pathway, but we now know there's a lot of genes. They never make proteins, they make RNA molecules, and the RNA molecule does a job. So, and then a chromosome is a large linear DNA molecule, and it has lots of these segments. The average chromosome in our species has about 1,000 genes linked up in a row, so to speak. And when we use a gene, that's called gene expression. It's an important concept to realize. A simple example, insulin is a protein, a small hormone, and you have to have a gene for it. So there's on some chromosome, there's a little segment, and it has the instruction how to make insulin. And the thing is, this insulin is only made in one cell type. Our body has at least 250 or more cell types. It depends how anal you are to kind of define them as separate. So there's only one cell type in the organ called the pancreas that uses that gene. No other cell in your body makes insulin, even though they could, because every cell has the same genes. Because all your cells come from a fertilized egg, so all the genetic material is identical in all the cells. So now you can see there's a big problem for nature. You need to regulate the gene expression that every cell produces the correct proteins. And that's a big job. The first step in gene regulation is when the DNA is transcribed into RNA. So that's called transcription. And nature's pretty smart. That's where most of the regulation takes place. There's more to it, but let's leave it at that. So who does that? Those things we call transcription factors. So those are products of genes themselves. Where else would they come from? Most transcription factors are actually proteins. So again, you have a gene that makes that transcription factor, and that thing then regulates the transcription of other genes. Since we sequence our whole genome, in humans we have about 1,800 proteins who are transcription factors. Sometimes people compare them, you know, you have people that work with shovels and pickaxe, they actually do work. That's the structural proteins. And then you have administrators that tell others what to do. That's them. So basically they orchestrate what's happening. And look at this. We have only about less than 21,000 genes in total. So almost 10% are used to just control what's happening. And those are very finicky proteins. If you change just one base in the sequence that encode such a thing, it may not work, or it may work much better, or it may work less efficient. So it's very sensitive material. And so they can have dramatic effects on what we call the phenotype. Now, what is the phenotype? Your gene sequence, by the way, the genetic code is a set of rules that is used by nature to convert a DNA sequence into a protein that has been discovered in Madison, Wisconsin in the 60s. You do not have your own genetic code, like some people say. That's a wrong use of the term. You and I, we have our own genome. That's your genetic sequence, which is unique. It's not your genetic code. So your genes, that is your genotype. Every human is different. Even identical twins are not the same because there's random changes as they get older. They're very similar, but they're not the same. And how you look like, that's your phenotype. That's also a wishy-washy term in some way because I can look at you, that's how you look like, but I can't see your blood type, can I? In a laboratory, I can. That's also your phenotype. So there's a lot of phenotypes. I need tools to figure it out what it is. So now, what about evolution and genes? Genes are obviously made out of DNA, and when you look at the DNA chemistry, it is impossible to copy them faithfully over long periods of time. So it is impossible, they must change. And those change, mutation merely means change. You know, when somebody calls you a mutant, that doesn't mean that you're weird, you simply are different. That's what mutare means, it's the Latin for change. So, and because of the imperfectness of copying, these keep making small errors called point mutations. And then the next generation copies that error and adds new ones. So it can be, it's impossible to maintain sequence perfectly. The downside of this is, in our own lifetime, if you keep mutating cells, and we do, take the colon lining, those cells divide once every day through your entire life. Do the math, how many errors they make over that period. That's why we have colon cancer. So in our personal life, mutations mess up important genes, it results in cancer. In the big picture, in species, those changes allow evolution. Usually when something randomly mutates, of course mutations are a random thing, things go worse. So that's why I call it devolution. Of course, then comes natural selection. What is not working usually gets eliminated by survival of the fittest. Another big change that is important in looking at this big picture is gene duplications. That's a well-established process. We know this is not well, it is a theory, but there's a lot of facts that say this is happening. It's actually shown that these are accidents in meiosis. When you make sperm and egg, it's complicated. I can't explain it to you. Believe me, genes can be, whole sections of chromosomes can become duplicated. And that's a luxury now because you have an extra copy of a gene. You don't really need it, but it's there like a spare part. And just like I said earlier, those spare copies cannot stay the same. They will, over time, over long periods of time, become different. And then they are often put to use by nature to something similar that's sort of new, but not the same. My only field of expertise, I worked for 11 years in research. I worked on steroid hormone action. That's an example of this. All steroid hormone receptors, these are proteins, they're transcription factors. For example, the testosterone receptor will regulate all the genes that you need to turn on to make a man. The estrogen receptor will do that in the female construction of a female person. So, and look what happened in evolution. We have receptors for estrogen, testosterone, progesterone, cortisone, vitamin D, vitamin A, thyroid hormone, there's a few more. All those receptors are very similar. It's like a car. Usually a car has 4 wheels that are black. There's at least one door and a window so you can get in and look out. These receptors have all those features where you can tell they're modeled after the same principle, but their specificity is different. And that's the result of gene duplication. Now, if we look at more down-to-earth things, what is different from us compared to our closest relatives, the primates? There's a list of more than 200 phenotypic differences, and I use here 2 extremes. No animal ever figured out how to use fire. Maybe that's a good thing. We had a lot more forest fires. And no animal has ever invented sports like ice hockey, right? So let's look at a few things in the limited time I have. How about the upright walk? No animal, no mammal, no primate actually walks all the time upright like we do. We know that from fossilized footprints like on the picture here, so it's been a long time. Also, we have anatomical indication by the location of the big hole in the skull. I'll show in the next slide what that means. The shape of the pelvis, you know, if you walk upright, the whole mechanics is different. Structure of the lower limbs, so we can reconstruct that for a long time people have been decided to walk upright. These series of pictures shows the hole at the base of the skull. Really, that's foramen magnum, means big hole. That's where the spinal cord connects to the brain. And the location, because of our heads sort of bobs perfectly above our heads, is centered. In a four-legged animal, it doesn't make sense to put it there mechanically. It's in the back. And in a knuckle walker like a chimp, it is somewhat off-center. And we have, of course, fossilized skulls where we see this has been going on for a long time that people walked upright. So then the skull is what's your head, right? And then the skull has a big part in our species and many others. It's called the cranium. The cranium is where the brain is housed in. And it doesn't take much scientific knowledge to see that the cranium in humans is much larger in proportion to the facial bones of the other primates. So here is the, that's actually the chimpanzee, that's the bonobo, and here are some of the extinct hominid species. So at birth, humans have a much relative larger brain size than other primates. The facial bones, as the image clearly sees, develop far less in our species. Human neonates, newborns, also are much larger in size compared to their mothers. Humans also have the highest fat mass. That's kind of surprising. There are animals that live in very cold places in cold water, like seals, but yet humans have the highest fat mass of all mammals. Purpose of this, I don't know. So I want to show you an interesting little aspect here. This picture shows the relative growth rate from a fetal chimp and human to the shape of the adult face and skull. And I use this example here. I used to teach pathology to the nursing students. So this woman developed a tumor in her pituitary gland that secreted an inappropriate amount of growth hormone. And my point of this picture is, so genetically there's nothing different with this person. Simply one hormone decides to be elevated and triggers some bones can grow. It's, again, a long story. Why can't you grow taller? That doesn't work. Your legs can't grow. But the irregular shaped bones in the skull and the hands can. And look what the genotype changes in this one person's life due to a little hormone change. Now imagine what can happen to you have 206 bones in your skeleton. If you start tinkering with genes that alter the rate in which they grow, you can modify the skeleton any which way nature wants to do it. Weird little obvious thing, when you look at humans, we can see the sclera of the eye. The eyeball is actually almost completely buried in the socket. You only see a very small part sticking out. And in no animal you see the sclera. They have a sclera. If you take the ball out, it's white, but you never can see it. And people are wondering, does that have a purpose? Why is it only in us the case? And some people have these theories. Well, that's for sure known. You can see much more clearly what a human is looking at than an animal. Does that serve a purpose? Probably in communication between people. And some people even go as far to say as when you're hunting, you can communicate to people by sight without talking, without making any noise. Of course, that's kind of extrapolating a bit. But anyway, it's a unique thing. That's all I'm saying. Unique thing, too, when you look at the growth of humans, so the red line is the chimp, blue line is the humans, we have this strange growth spurt in and around puberty that is absent in the other primates. Newborns, I don't need to tell you that, are extremely dependent on maternal care for a long time. That is very unique in primates and even more so in humans. Strange thing too, humans usually wean their children by the year of 1.5. You might say that's not right. I'll tell you later. Now the modern humans have a special adaptation because of the lactose intolerance thing. But naturally, Homo sapiens could not drink milk anymore by the time you're 1.5 years old. You would get sick from it. So then the baby doesn't even want to nurse anymore. So that's very different in all the primates. They nurse their offspring much longer than that. One upshot is humans ovulate much quicker, so they have faster reproduction, and they had a much faster population growth because of this. Now, this is published in a prestigious journal just recently in 2014. How much does it cost to operate and build the big brains that we have? So the brain's metabolic requirements peak in childhood when it uses glucose at a rate of equivalent to 66% of the body's resting metabolism and 43% of the body daily energy requirement. So our brain is in some way a very expensive thing to operate. Adults are not much different. An adult brain consumes 120 grams of glucose every day. That amounts to 480 calories. That's almost a quarter of the entire resting metabolic rate. My point is, like a dog has a very much smaller brain than us. It takes a lot of energy to operate such a big neuronal factory. Did I just click twice? No. Now, physical performance. Humans are the mammal. Of all mammals, we have the highest capability of endurance. You know, I don't think the old Neanderthals or Homo Sapiens would consider it they're exercising. They were working. They're trying to survive. It wouldn't be sports to them. So, and this has been traced back to almost 2 million years ago. People were able to do this. And the biggest adaptation that humans have to allow endurance performance is thermoregulation. We all know when you move, your body muscles create a lot of heat that you got to get rid of. And they figured out how to do this. Loss of body hair, and then the development of sweat glands. Even today, you have eccrine and apocrine sweat glands. It's those eccrine sweat glands that make a very watery secretion with sodium chloride in it that are very good at cooling the body surface. Here's an interesting twist to this. We crave to eat salt. And that must have an evolutionary advantage because sweating causes salt loss. And so salt wasn't easy to find in most diets. So it was very good if you have it, eat it. In today's society where we have fast food and we mine salt and it's everywhere, that has a downside to it, as we know. So a lot of people get sick from eating excess amount of salt. Now, shedding your fur coat has problems too. So very little short body hair. Uniquely in our species, we have hair that grows on our heads quite long. Again, very complicated science, those follicles, how long can they grow, blah, blah, blah. It's unique to our species. When you want to study a lot of the stuff, the devil is in the detail. When you look at the structural protein of hair, is called keratin. Well, you don't have one gene, you have 30 different ones to make hair. And we don't even know yet, why is hair curly, why is it thick, why is it thin? Somewhere it's buried in this. And then add to the mess, there's another 100 keratin-associated proteins that determine how and which way hair grows. So it still elusives what the real difference is between the monkeys and us. Now, the body hair loss was very early, and it allowed that thermoregulation during the hunter-gathering lifestyle of people. And then you create another problem. Once you have no hair, and if you migrate into more northern climates and you deal with cold, you need to figure out how to build clothing, shelter, how to control fire to solve the problem of cold. Now, what about once you have no fur coat, you're vulnerable to the exposure of UV light. So then what about skin color? Obviously, we know today we have a big spectrum of human skin color. How did that occur? When you look at a map of the globe, the coloration shows where people live that have the darkest complexion versus the fairest complexion. And it very much coincides, of course, with the sun exposure. This picture here is a UV index map of the world in October. And it's for each location done at noon. So when you look at, as you know, in the equator, it doesn't really matter what time of year you have. It's always summer for them. So the UV intensity is incredible. It reaches sometimes 20. In Canada, even in the summer, we might hit 8. So the UV light is very, very intense in many places of the globe. So now, what about skin pigmentation? Now again, you might have learned Mendelian genetics in school or in university. Good old Mendel, he was looking at purple and white flowering pea plants and crossed them. Wouldn't you predict that if you cross a purple and a white plant, that it somehow becomes intermediate? Any common sense would tell you that, but it didn't. So what's going on there was what's called dominant recessiveness. Most genetic traits are not like that. Most are actually called non-Mendelian traits, and they often are quantitative. So and what that means, you take a very dark person, so that person's genes contributes very darkness to the skin, and you take a very light colored person and you get exactly what you predict. You get an intermediate color. And there's actually many plants, when you do this experiment, that's exactly what you get. Take a red flowering plant and a white, and you get a pink one. So that happens lots of times. So the molecule that gives us skin color is called melanin, and it also stains the hair and is partly of the iris. And in dark-skinned people, we have many large melanosomes. These are the organelles where melanin is made and stored. And the melanin they make is called eumelanin. Chemically, it's different from the fair-skinned people. So fair-skinned people, of course, they have the opposite extreme. They have few smaller of those melanosomes, and they make a chemical different called pheomelanin. So now the primates, other than humans, actually, if you shave a monkey, they're white. They don't need UV protective skin coloration because the fur takes care of this. So they have some parts that are pigmented, but for the most part, they're not. So now when people shed their hair for thermoregulation comes the new problem with UV exposure. And UV exposure is actually very dangerous. The UV light around this wavelength here is just banging up DNA molecules, proteins, lipids. It's very destructive. Hang clothes in the sun for months on end, and you'll see how colors break down, right? It's very harsh. And so that is now big pressure to adapt some sort of protection in the darkening skin. Just to give you a quick fact how dangerous acute sunburn is. So erythema, the reddening and blistering of the skin, that will cause permanent, if it's severe enough, will cause permanent damage to sweat glands. After that, you have problems with thermoregulation. Severe sunburn can be immediately fatal to people. Then there is a more subtle problem. The UV radiation can damage a lot of nutrients that circulate in blood in the skin. And so that would affect flavins from plant food, carotenoids from plant food, and vitamin E and folic acid. And that's a big problem. Skin cancer probably is not important for evolution because that takes time, and it's probably for the development of the species not very important. For us, of course, in real life it is. Now, natural selection. So once more, this is a quantitative trait, the skin color. And there's a very strong pressure against mutation away from dark skin if you live in the tropics, because you have bad genes to get all these nutritional problems. There's a very weak selection once you are in higher latitudes where this UV exposure is a lot less. So there's a lot more diversity in the skin coloring in more moderate UV areas in the globe. Now here's the extreme. Shannon, I used Prince Rupert and South Sudan as two extremes. So a dark person is perfectly suited to live in a very sunny place. They don't get a sunburn. Their micronutrients are protected, and despite of being so dark, there's so much UV light, they make enough vitamin D. Now send a very light-colored person to the Sudan, and you have big problems. That person gets burnt to a crisp. You have nutrient deficiency problems, like folic acid deficiency is to blame for neural tube defects, like spina bifida, and other problems. They can make lots of vitamin D, but that doesn't help. Send a very dark-colored person to the north. Sunburn micronutrients is no problem, but now the absence of vitamin D becomes a big problem. A person with dark skin in Prince Rupert without supplementation will guaranteed, if it's a child, develop rickets, if it's an adult, osteoporosis, for sure. So now, I don't have enough time. Today we know at least 13 genes that contribute to the skin color. I'll just give you one example, this so-called KIT ligand. It's a protein that determines how long those melanocytes survive and how much they proliferate and how they migrate. And all it takes is in position number 326 of that gene to change an A to a G, and you have a big change in skin color. And this has happened. So do you see how little it takes to make a switch that explains in the current populations between the African and non-Africans about a fifth of their coloration. And once more, every gene in your genome you have twice. One from your mother, one from your father. So you would have 26 versions in total of, they could be dark genes, they could be light genes. And when you make an egg or sperm, you put only one of each. So you would put 13 copies, and your spouse 13 copies, and you have a new combination. And that gives you not endless, but a very large combinations. That explains why there's such a spectrum of color possible. Now, interesting, hand preferences. No other primate shows preference of right or left-handedness. 90% of humans are all right-handed. It's all just facts. Lateral asymmetry. So when you look at the brain, even your body, your heart is on one side, right? And your liver on another side. So there is a certain asymmetry in a lot of our structures. And it's all genetics. There are people that are exactly backwards. Their heart is on the right, the liver's on the wrong side. That's all possible. It's all genetically determined, including the brain. The Neanderthal already were right-handed. People know that from looking at the shape of tools and tool marks. So it happens at least 500,000 years ago that people became preferred right-handed. Also, the language centers in the brain is asymmetrical, as I'll show you in a sec. Already in ultrasounds, you can see pictures, fetuses sucking their thumb. And when they suck their right thumb, they turn out to be right-handed. So it starts very early. It can't be conditional that we say people don't use your left hand. It's biologically. And of course, 10% of people are different. They're left-handed. Now the cortex too, our brain structures. The left hemisphere is in charge of language skills, mathematics, logic. They sometimes call that the science brain. And the right hemisphere is more visual, spatial skills, intuition, emotion. Some people call that the arts brain. Of course, that difference is not to be understood as one dominates over the other. Of course, they cooperate. But the point is, it's not symmetric, it's asymmetric. Down to the wire, even when you study gene expression, I don't know how the heck they did this, study gene expression in the language areas in fetuses. How old were those fetuses? 12 weeks. I try to find this paper's material and methods. I don't know how they got a hand on these human fetal brains. But anyway, here's what they report. Harsh difference. So here's a number of genes that are highly expressed in the left hemisphere, and then very different ones on the right hemisphere. So that must have some purpose. Again, gene expression is the story here. The human diet. Humans, of course, figured out how to use fire, and we're the only species that can do that. Cooked foods have a lot of advantages. It's funny, sometimes people say fresh foods or raw foods are healthier. Yes and no. We're better off with both. There's a lot of advantages. You can get a lot more energy out of lots of plant food when it's cooked. There's a lot of chemistry going on when you gelatinize starch, denature proteins. Simply things become more digestible to a human. Also, there's a lot of compounds that are toxic until you cook them, then they're non-toxic. A lot of food naturally is loaded with things that make you sick, bacteria or fungi. A lot of those, when you cook them, they become, well, tolerable to us. Also enhanced flavors, obviously cooking things, barbecuing things, pickling, etc. We agree with the flavor. In fact, I don't need to tell you that most animals, if you give them the choice, they would sit at your table and they would not eat rotten salmon, right? I bet you. So also cooking food makes us spend a lot less time foraging and chewing on materials. Anyway, let me carry on here. Let me show you a more fascinating recent thing that happens in our diet. And that is one of the, actually scientists say this is the strongest point of an example of adaptation and human survival because of one simple gene change. So lactose intolerance was the norm for Homo sapiens. People could not digest lactose in milk once they're past infant stage. So after weaning of breast milk, adults did not consume milk. Where would it have come from? Unless you have cows and other animals. They didn't. So there was no point. And by the way, that would make a lot of sense. Why would you make a protein? Lactase is an enzyme. Why would you make this if there's no purpose for it? So they didn't. And it's actually bloody complicated to turn this gene's expression off. So it was expressed in infancy. And then, by the way, similarly, you made a fetal hemoglobin when you were a fetus. You had to steal oxygen from your mother through the placenta. It's very different from breathing air in the lungs. So as an adult, you use a different gene to make globin. Same thing. So now, what happened is there was a point mutation in the region of the gene that regulates this shutdown in childhood. And so now the gene is expressed. If you can digest milk, you're one of them. So now the gene is expressed throughout life. And we know what these mutations are. There are 6 known today. There probably are more in some corner of the woods, the world. So 2 of them occurred in Northern Europe and then in Central Africa about 7,000 years ago. That is not long in terms of evolution. It's very recent. So in Europe, when it says minus, this position is 13,910 bases of the gene upstream of the start of the gene. Genes are transcribed in one direction, that's the downstream. If you go in the opposite direction of the chromosome, that's upstream. So the mutation is 13,000 some bases away, and there's a C switched to a T, that's all. And that has messed up the repression mechanism. It's actually very easy to break things. It's very difficult to make it. So where this came from, I don't know, but we have broken it some 7,000 years ago. In Africa, in Kenya, it's a different spot. See, it's not the same spot, different mutation, same effect. It's part of the mechanism. So the African people that can drink milk have a different reason than the Europeans. The outcome is the same. In Sudan, there's another one, and then in the Middle East, there's another one. So there's probably more somewhere in some corner of the earth. Now, what's the upshot of natural selection? If you have milk-producing animals, and you have a nutrient that the ordinary humans couldn't use because they would get sick, they wouldn't want to have milk. They got diarrhea and all kinds of discomfort. But in a pinch, in a famine, those people would have the edge to survive because of the access to a nutrient. Milk is very good. It has minerals, it has protein, it has fat, it's water, it's sterile, it's clean. So, and there are some areas in Europe, Finland, Hungary, there's almost nobody left with the original sequence. They're all lactose tolerant. So there must have been a major selection in those 7,000 years where the original Finns and Hungarians, they're all gone. So the mutants are surviving. Here's a map. By now it's outdated, but it doesn't matter. My point is, the Native Americans who haven't interbred yet with immigrants are all lactose intolerant. So are the native Australians. They're all lactose intolerant. A lot of people in East Asia are all lactose intolerant. It's never had that mutation. Of course, migration, that's why it's all blue here, that's Europeans that brought that mutation over to the Americas and to Australia, of course. Now, one more quick thing I want to talk about. I should skip this here. I have too many slides here.
That's okay, there's no rush, we have the room.
Okay, well, tell you something about human amylase. So in your saliva, you have an enzyme, again, that can digest starch. And so most human ethnicities have more than one copy. Here's that gene duplication at work again. Some have 20 copies rather than one. All apes have one. So somehow gene duplication occurred, and then it became an evolutionary advantage. So the invention of the dairy allowed the lactase mutation to thrive. The invention of agriculture by eating tubers and rice, which apes don't, gave us the advantage if you have more than one amylase gene. So the salivary amylase, you might think, I don't know, do you think about when you swallow, the salivary amylase protein might die in the stomach because it's so harsh and acidic? Well, there's enough that survives that it actually works. Not as effective as it does in your mouth. Especially now if you have 2 enzymes, genes, and 4 and 8, all the more amylase will survive and go to work to digest your starch. So it does work. And so here's a picture, again, from that textbook I mentioned to you earlier. It shows, first of all, the original gene, which is persistent in all the monkeys. And then in humans, we have multiple cases of duplications to the point that some have, there's 1, 2, 3, 4, 5, or up to 20, like I said. And what's compared here is the Japanese population that has a high starch diet, and the Yakut people in eastern Siberia that have a very animal-based diet with very little starch. And look what natural selection does. This is the allele frequency. So what it means, only 9% of the Yakut people have lots of amylase genes. Nobody has this number. Many have this one. 63% have only 2. You go to the starch-eating people in Japan, so many more people are represented in the high number of these gene copies. So that's just a fact. And somehow, how do you explain that? There must have been natural selection favoring one or the other. Now, what about our brain? My favorite character, Homer. So what is special about our human capacities? I know a lot of people don't like accordion music, maybe, or banjo music. You know the perfect pitch? When you throw a banjo into a dumpster and it hits an accordion on landing. I can play the accordion a little bit. It's goddamn complicated. This is like a piano, but those buttons here, you push one and it's a C chord. You push another one next to it, it's a minor. You push another one, it's a 7th chord or a diminished. So would you imagine how the hell that thing works? All the pipes that run in there and then the bellows that blows air. Somebody was thinking about what they're doing there to invent such a complicated instrument, or build a big pipe organ or a big bridge. My point is, biologically, we're not that different, but neurologically, I think we are. So we have incredible mental capabilities to accomplish that sort of technology. So it's funny, sometimes I've read lots of textbooks about evolutionary biology, genetics, etc. And often we are downplayed. Some people think humans are arrogant. We consider ourselves special. And so they want to say, we're just another animal. We're just another primate. Well, that's sort of downplaying those facts. Monkeys don't compose and play music. They don't invent funny sports like skating around with sticks or doing other complicated things. By the way, athletics is a prime example of an incredible required brain power, because you need to control your dexterity, your balance. This requires enormous computation by the brain, especially when you're walking upright. Ask anybody who builds robots that's supposed to walk, In difficult terrain, it's very complicated to not tip over because you need adjustments all the time. It's what our cerebellum does. So then we can, of course, have language. I have to say about this. We can do math, build airplanes. You get the idea. We can do science. And more on the practical side, we figured out how to protect us from cold and weather. We grow and secure food in agriculture. We grow barley and wheat for entertainment. Little pun intended, right? We even engineer food. We synthesize and isolate medicine. So obviously, no animal comes even near this kind of mental achievements that we can do. So now let me tell you quickly something about the brain. It's still a big-time black box. We know where a lot of stuff happens, but how it really works, we really don't know too much about it. So what's shown here in these colors is the cerebrum. And the cerebrum is where all the sophisticated stuff is going on. And the cortex, like the outermost part, is where all the neuron bodies sit. It looks gray when you slice it with a knife. The white matter is where the projections go, the axons. So the cortical mass seems to be the most important for all this mental stuff. We have, literally, that means small brain. So there's another brain attached here. It's called the cerebellum. And that is majorly involved in motor control. Now, here's some numbers. So the functional organization of neurons. The entire nervous system in a person has about 85 billion cells. That's the neurons. But look at, that's a number, well, lots of cells, right? No big deal. You have lots more white and red blood cells. But look at this. Sensory neurons are the ones, these are wires, more or less, like electrical conduits, that send information from the periphery to the brain. Now that would include all your sensation that you can feel, your temperature, pressure, vibration. Then, of course, all your senses, hearing, smell, taste, vision, balance. All of this internal organs, like you send information on blood pressure, on heart rate, tension of the intestinal organs, bladder. All of this information is sent to the brain. How many wires do you need to do that? 10 million. There are only 10 million wires sending all the information to the brain. And look at how biased that is. A quarter of that comes from the eyes. The eyeball itself has 1.2 million axons, and the optic nerve is almost the thickness of my finger. So all other nerves are actually much, much smaller than that. So fine, 10 million. Those numbers make only sense in what I'm trying to tell you when you see them all. Now, what about outgoing stuff? So motor neurons send commands to the outside. For example, you have 600 or so muscles. They need to be told what to do. You have all the internal organs. The heart is controlled, bladder function, secretion of glands. So there's lots of autonomic nervous system and motor system that is outgoing. How many of those do you need? What would you guess? So 10 million sending information. How many do you might need to operate the action? Give me a number. 40 million? 500,000. For example, big muscles in your leg, you have several hundred muscle cells. They have only one axon, which branches and operates them all together. You know, when you kick, that's a pretty crude action. You don't need fine control. If you work with your fingers, there very small number of axons control small number of muscle cells that have fine control. So, but anyway, it's very small, that number. And look at now, what are all the other neurons? So an intraneuron sits somewhere here. It doesn't project out. It doesn't tell anybody what to do. It doesn't listen to anything coming in. They just talk to each other. So hence they're called interneurons. Their axons often are very short, just a few micrometers, talking to their buddy next door. And what are they doing? Number one, you have 20 billions. Look, I put B here to stress the point. That's 1,000 times plus some more that does all the computation. So this is where interpretation, planning, coordination of signals going in and out, that's where the decisions are made. And that's the part where we understand very little. What the heck are they doing? How can they learn? How can they form memories? There's something in your head tomorrow that wasn't there today that we call memory. What happened? We don't really know. We know it involves gene expression because in animals we have drugs that block transcription and translation, then they can't form memory. So there must be something at the very guts of how cells work happening. We don't know. Now, language is the most fascinating thing as far as I'm concerned about humans. People speak thousands of languages. Many, of course, went extinct, and it's still going on, of course. Many languages, I'm not an expert in this, that's a linguistic is a whole science that I read something about. They tell us there's lots of shared characteristics. All human societies have language. Groups with simpler technologies and organizations do not have simpler languages. So you don't have to have a high culture to have high level of communication. They all do. Most individuals in a society are competent users of language. Some are really clumsy at sports, right? My mother would say they have 2 left hands. But most people can speak. They can communicate. Language is innately acquired. Also fascinating. If you were sent to China, being born in Canada, at birth, you have no problem learning Chinese. It doesn't matter. Any person, barring some brain damage, can learn any language in childhood. Now, do we have fossil evidence? No, we don't. But people in this field, Shannon, you can prove me wrong, in archaeology, people often have deductions of when they see this, they say this must have explained that. So for example, tool use and manufacture is, by the way, also biologists say, oh, animals use tools. Yes, they do. Animals communicate, but they don't have what we really call language. They communicate, but not at the level we do. And tools, if you have a stick and you poke it into a hole to suck out some termites, that's a little different from the accordion, right? You get my twist, right? We're far more complicated. So, and here's the deduction of this. You would require sophisticated language, teaching language, most complex skills, whatever it is, playing an instrument, doing eye surgery, whatever it is. You need periods of long learning practice, and that needs instruction. I've read the book Guns, Germs, and Steel, and Jared Diamond explains how long it took the Spaniards to figure out how to make swords that wouldn't break. They could be sharpened, and they would be flexible enough not to shatter. Centuries. Don't you need to explain to your offspring what you did that didn't work and what worked to improve this? So without language, you would have no culture like that. It's impossible. So, and also there's a very practical thing. If you can communicate with language, you can talk to lots of people. Next thing you know, you can have clans, tribes, villages, cities. You can have cooperation between very large number of people, and you become very successful in competing with other animals. So cooperating in hunting and gathering is an obvious, immediate survival advantage. We became top of the food chain by cooperating rather than being eaten and devoured by a big beast. So, and you need a lot of brain power to control speech. There is the perception that you can understand spoken, sign language, or even written language. That's one thing. And then there is the ability to produce language. Unfortunately, we know lots of all this from victims of strokes who had a very small, a stroke is usually a blood clot in a very distinct area of the brain, and that area will die. And if that area was necessary for the motor control, you might still understand language, but you can't formulate words anymore because you're lacking the outgoing power to do it. So, and there's lots of genetic diseases also known that cause problems in those areas. There's one interesting gene that is required for language called the FOXP2. That was actually discovered in the British family. It's a spontaneous new mutation. Those people can't produce verbal language. They can't speak. Very severe dyspraxia, it's called. And also they have impaired learning. They have a poor lower mobility of their face. They have all kinds of problems. They have poor language perception and grammar understanding. And it's just one single gene that is not working, causing all this. And that's a very interesting gene. It is a transcription factor. So it actually helps forming parts of the brain that seem to be necessary for language. And when people say genes are preserved in vertebrates, that's a big group of animals that have a vertebral column that includes, of course, all the mammals, includes the primates. Something is important if it's found in that many different critters. So the FOXP2 gene, it turns genes on that are involved in vocalization in all vertebrates. It's highly conserved, so the sequence is maintained. Probably when you change it, that guy dies because that was a bad idea, so you need that. In songbirds, for example, it's expressed during the period when they learn how to sing. It's funny, there's not every bird, like a chicken is not a songbird. They just make some noises, but it's not sophisticated. These little birds, if they have that gene expressed at a right time, they can learn from their parents how to make their songs. And if you take them away from their parents during that time, they miss that. They never learn how to sing. They're quiet. We can make transgenic mice. There's a company in England, they have knocked out every one of the 20,000 genes in mice and see what happens when it's wrong, when it's absent. Those mice without FOXP2 genes, they're quiet. They don't make any funny noises that mice can do. In humans, that's the speech impediment I already mentioned. So this protein, the FOXP2, is a protein, and it differs from the chimpanzee by 2 amino acids. The Neanderthal had the same sequence as us. So at least genetically, they probably could speak, but anthropologists are not sure whether they had all the rest that you need to be able to speak, but they had that gene. So the differences scientists in this field tell us are definitely part of the reason why chimps don't talk and humans do. Now here's an interesting series of images. These are PET scans of a person. You tell a person, close your eyes, and they're in the PET scanner, and then you give them a sheet of paper with a word on it. Then you tell him, open your eyes. And if he does, the optical nerve projects to the occipital lobe. That's where the visual perception area is, so it becomes active. So he sees the word. Then comes the area where that word means something to that person, because obviously you can't use that with a Chinese word for an English speaker because he doesn't know what that means. So you need to give him a word he can understand. Then the Wernicke area becomes active, and if you ask him, say the word, you can see the premotor area, that's the planning of speech, and then the motor cortex. It's a very small strip on both sides that coordinates then the actual movement of the muscles. You know, you need to move your tongue, your cheek, your larynx, your what's that thing called, the diaphragm. So all these muscles need to be told to act properly to speak. And so those areas are well known, so the sensory area and the motor area and so on. So how does this develop, and why don't other animals speak like us? Neonates, like newborn humans, they can make noise, they can cry, right? They can cry very loud, but they can't speak yet. When they're about 1.5 years or 2, suddenly they can really take off. I have grandchildren now. When I had my own kids, I didn't really pay attention to it. They talk. But when you're older and you watch them, and very quickly you realize, oh my goodness, how quickly do they pick up all these words? And next thing you know, they start developing grammar. They know how to make sentences that make sense, the right syntax, and so on. And I observed this myself, that they use logic rather than real language. Like they say, I put it in there. Of course, it's wrong English. Or, look, there's 2 deers. It's logic what they say. The brain works fine. Just the language that has culturally evolved says, no, we say, I put it there, right? Anyway, so very quickly they learn this. And when you look at I don't quite understand this method of brain imaging. It's a very modern thing. So what they've done here are these images of the left and right hemispheres. So it's asymmetric, the speech center, in a newborn and then in an adult. And the key is that the Broca's area is missing those blue connections that form somewhere between newborn stage and adult. In fact, it forms right around the year of 1.5 to 2. That's exactly when children start to develop real language skills. And so in a different publication of the same scientists, they compared brain imaging in primates, like the macaques, a chimpanzee, and a human. It's a very complex story how this all works. In humans, there is a real circuit of those centers that forms when the child is about 2 years old. That circuit never forms in any other animal's brain, not even in the chimpanzee. So somehow there's a small connection where communication would have to be processed. They don't make that connection. Now, interesting, Alfred Russel Wallace, he was a contemporary of Charles Darwin. In fact, he was part and parcel of the evolutionary story of the origin of species. And he had some doubts whether natural selection can explain everything of survival of the fittest. And language and the mind was his biggest stumbling block. Here's what he said. Man passed on innovations in the form of mathematics, art, music, moral teachings, etc., and did so regardless of whether there seemed to be any apparent survival value involved. How could organic evolution proceed in a fashion responding to environmental constraints and opportunities, yet produce adaptive structures whose persistence was not necessarily a direct and exclusive function, those constraints and opportunities? What he is questioning, we have come to a stage of our brain development. We can do stuff that is of no use for survival. Why can't we do it? So that was his question. By the way, if you're interested, so Why Only Us? That's a book very recently published about human language. That's where some of the information is from. If you Google Noam Chomsky as well as Rob Berwick, they have lectures about this whole book online. You can watch them for free. Very fascinating. They have a lot more to say about the whole story about human language. And, well, the title says it all. Why Only Us? They are convinced that's a huge difference. Yes. Now, let me just summarize some of the genetic findings between us and other organisms. So once more, many genomes have been sequenced. The chimp, orangutan, gorilla, bonobos, human species, of course, Homo Sapiens. Thousands of people have been sequenced. And then, of course, a couple of the extinct hominid series. Now, the genome size, our size is about 3.2 billion base pairs. And the human and chimpanzee are genetically the closest. We match about 98% of the sequence. And interestingly, we have 23 pairs of chromosomes. The monkeys, all of them, have 24. And so that seems like, oh, what a big difference. Not really. What happened is human chromosome 2 is a fusion. All the apes they're numbered now, their chromosomes after us. They have 24 chromosomes, but what happened is chromosome 2A and 2B are fused into one in our species. So all the genes are just on 2 chromosomes that we have on 1. There's lots more evidence that that's happened sometime when we split from them 5 or so million years ago. Now, since we sequenced both of them, the computer says there are 14 million differences between chimps and human DNA sequences. 35 million are single base changes. So that's those little errors that happen when DNA is copied. Another 5 million are other errors where a base has been lost, deletion, or one has been inserted. So lots of changes. Now, what does it mean? The vast majority are neutral. Maybe I don't have enough time, but maybe I can tell you. Of those 3.2 billion letters in your DNA, only 1.5% code for proteins. So you can have a lot of bases change with no effect, because it's like shooting at a house trying to hit a window, and 98% are not windows. So you will not hit anything that's of relevance. So there's irrelevant bases in introns and repeat sequences there. They don't matter. So those cause no change in any amino acid in a real protein. And also in biochemistry, we learn some amino acids, when you change them for another one, it works just as well, because some amino acids are very similar. Complicated. But some account for phenotypic differences. So the actual structural difference in the gene product or RNA matters. Or, for example, I gave you some examples. Changing one base can make a big difference in how much a gene is expressed because that base was important. Now, 35 million single substitutions. What's the upshot of all this? 100,000 are actually in exons that matter. So exons are the parts that really code for what becomes a protein. 40,000 of those are amino acid substitutions, so there's something different. 20,000 of those substitutions are human-specific. That's an important thing to think about. All the monkeys have the same sequence, but the humans have a different one. So there's probably a reason for that. What it is, we don't know. Nearly one amino acid in every human protein is unique, not found in any of the apes. What's the functional significance? We don't know yet. You need to study the performance of every one of those proteins to figure out what might be the impact on brain development or anything else in the human. Studies like this have been done in other organisms, and from that we know usually it turns out that 80% of single amino acid changes didn't really matter much, but that leaves 20% that do. And 20% of what we started out with is 4,000 proteins. So probably what is happening, we have 4,000 changes in human proteins that could have the key why we are so different from the other apes. We don't know yet what that is. So also we have 23 gene duplications in humans that are not found in the apes. What's the point of that? Let me finish here. Evolution rarely invents new genes. Usually they just tinker with what's there and modify it slowly. Often they were duplicated in the past. Related genes are found, these are computer programs. It's actually fairly straightforward. If you sequence a monkey, let's take a mouse. You sequence a mouse and a human. At first, all you have is billions of letters. It means nothing. But you feed this into a computer and you ask the computer, tell me where you see stretches that match the two animals. After all, they're vertebrates. They're both mammals, so they must have parts in common. The computer will then tell you, I see a sequence here where we have 98% match. Here we have one that is 75% match between the two species, and then there are big seas of junk where they don't match at all. Those blocks that show high similarity, that's the genes. That's how you find them. But if one species doesn't have that gene, you can't find a new gene because there's no match that it can find. So new genes have no sequence similarity by the nature of that they're new, and they're hard to find because there's no computer program that can identify them. So, but there's other methods. People have found new genes. Here's a list of a few of them. See, the number is very small, but remember, they may be transcription factors. They can have huge implications. I know from my steroid hormone field, a lot of transcription factors in this field, they end up regulating 500, 1,500 different genes, like dominoes. So who knows what they're doing? And look, most of them are found in the brain and not so much in other organs. Now, one last thing. By very carefully comparing sequences in all these genomes that we have, people found something they coined in 2006. They named that human accelerated regions. Now, what that is, there's only 49 such sites that were found in our genome, and a quarter of them are genes that have something to do with brain development. And so what this is, I'll show you the numbers here. When you have a sequence, this HAR1, that is the same, that region has only 2 changes between chimpanzee and chicken. So chicken, that would be mice and dogs and all kinds of cats in between. That sequence has been conserved over long periods of evolution of 300 millions of years to stay pretty much the same. It must be important, or else nature wouldn't have done that. But we have gone to work. We have 18 changes in our little path of evolution in that little region. What the heck does that do? We don't know yet. But it is a gene that is involved in structuring the neonatal cortex, the brain cortex. So And by the way, there's 48 others that are identified by the same method, so that's only one of them. So in conclusion, most unique human characters definitely affect the brain. Most of them are very subtle genetically. That's why we don't see like a big difference in our genome. It's hidden somewhere in those subtle differences. Probably there will be few really new genes that we haven't found yet. More likely it's just tweaking what's there that explains all the advantages that we have in our brain. So most will probably have to do with differences in gene expression levels. Look, I have one funny picture. When you read that book, I recommend Why Only Us. He makes this point. You know the story about Red Riding Hood? The wolf has come in and eaten the grandmother. And now the wolf is in bed with the grandmother's clothes. And so these authors are making this as using it as an example how sophisticated little kids' comprehension of speech is. It's because it reads in the story, Little Red Riding Hood thinks that the grandmother is in bed. So that sentence actually communicates something very complicated. The kid knows that the grandmother has been eaten because that was part of the story. But at the same time, it already comprehends from the mother's reading that the truth is really not what is portrayed as like or probable or possible, but that there's actually a distortion of truth. And to communicate this, how much brain power do you need to understand such a subtle thing? And little kids at the age of whose parents? How old do you need to be to understand that story? That guy's too small. He would eat the book. But let him be 3 years old. I bet you he can understand that. So, and that step never happens in any animal that can put that kind of logic into a complicated structure like that. Anyway, so we have to stay tuned if scientists find more answers to the uniqueness of humans. And thank you for listening. I'm happy if I can answer any question. Yes?
Well, I have a couple of questions. One to do with affect and how that influences or how that is expressed.
What was that?
Emotion.
Emotion, yes.
Yes. We didn't talk about it really.
Emotional weeping is actually one thing that humans can do.
Yeah.
It's not found in any other primate.
Yeah.
I can tell you one more thing. I deleted lots of slides. Schizophrenia literally means split mind, is a condition where there's no comparison found in any animals. And so that's another property. It seems that it's a bad disease. It's very common, actually, and it's highly genetic, highly inheritable. Doesn't, again, follows Mendelian clean slates of development. It's a very combinatorial thing. But the inheritability is very high. And some people say having such a powerful brain comes at the cost of having mental illnesses because it can backfire this whole system.
It's the curiosity of, how feelings function. I mean, there's a lot of stuff happening in the body when you're experiencing anger or regret or something like that. And the relationship of that to, just the lockstep science of this protein does this and so forth. It's much more complex. I think emotion has linguistic parts to it at times, or maybe it's merely the brain interpreting what's going on in the body and naming it in various ways. Anyway, that seems to me that's something that gets overlooked sometimes.
Very poorly understood. Can I just make one comment?
Yeah.
How little we know about the brain. Take something like migraines.
Yeah.
We have one word for migraine, but I'm a pharmacologist. We have so many drugs that physicians have tried. They call it off-label use. Like beta blockers were invented to treat heart disease. And when you have people with untreatable migraines, sometimes they try anything because nothing works. And lo and behold, sometimes you find a drug, like a beta blocker, that helps somebody with their migraine. Another it doesn't. Another takes a serotonin blocker that's an antidepressive drug, helps somebody with migraine, not another one. What does that tell you? they don't have all the same disease. There's something different, but we don't know what it is. Same thing with depression. Anxiety is a bit simpler. Most people's response to anti-anxiety drugs are similar, but with people with depression, same thing. There are some people, they respond to this drug, and others don't respond at all. And then there's other drugs where it's the other way around. So obviously, they don't have the same problem, or else they wouldn't respond differently to a drug. Don't know.
Skin pigment change with ultraviolet, is there any chance that could go backwards?
Oh yes, it has.
Yeah.
Didn't have time to talk about it. So humans had to go dark because of the loss of hair. And then when they started migrating north, finding new territory, gradually the pressure was on to select for lessening pigmentation. But then subsequently people migrated into East Asia, back into places where it's again very sunny. So actually people have gone through becoming white and becoming dark again. It takes such little changes in the DNA sequence to cause these changes that it's easy to follow that that's possible. And when you look at the very dark-skinned East Asian, their genetics is different from the very dark-skinned Africans. You can Google it, there's a website everywhere.
I'm glad there's not a test.
Also, of course, this is genetics. All those races can also tan. So that's another adaptation which we all have. Like, obviously, if you're constantly without clothes outside, summer doesn't come by flicking on a switch. It comes gradually in the spring. So, and the adaptation to more sunlight simply occurs when it's actually bloody complicated. It involves DNA damage and a response, and then you start making more melanin, and you know that people can tan quite intensely, but it's sort of slow. And then you need to also lose that tan, or else again you lose the ability to make vitamin D. So the dark-skinned people have that on top of their already dark complexion, so it's very good. Oh, there was a question?
I'm interested in the hand dominance.
Yes.
What is the survival advantage of being right-handed, or was the dexterity on the left side sacrificed by the development of the right side of the brain?
I've never read an explanation whether it is. It might have been a fluke. I've never read a reason. In fact,
90% of us, that's just stunning.
Yeah.
I have never read an explanation, but it may be simply a fluke that, yeah, I can't tell you.
Further to that, to do with musical instruments, you were talking about, the accordion. But if you take something like playing the piano, which requires quite a bit of ambidexterity, because the hands are, they're mirror images, but they're doing similar things. Are there any people who are born ambidextrous, as opposed to the one side of the
I don't know. I heard a nice quote. Any scientist who ventures out of his field of expertise is just as dumb as anybody else. I know only something about a very small slice of science. In fact, when you deal with stuff like this, I have to venture into all kinds of areas like linguistic. I don't know really much about it. So I can only educate myself and put it all together, and hopefully it all does make sense.
I think that's a nice spot to close on. Thank you, John. I think we're all interested in who we are and finding our place in the bigger picture. I do have a small token of our appreciation. Thanks for once again giving us a very stimulating talk. Thanks, John. So I hope we see you all a couple of weeks from now, even though that's reading break or ski break, as they call it here on campus. We'll be here and we'll be having a presentation on the 20th.